Thanks very much, Davna, and thanks to the GMBA for hosting this webinar series and inviting me to present some alpine plant ecology from Australia. I live and work on Wurundjeri country in Naarm, which is the Aboriginal word for Melbourne, and I also work across the countries of the Yatmatang, the Gurnai-Kurnai and the Garigo country people, and I'd like to pay my respects to the elders past and present of those groups and acknowledge that there's still no treaty and no sovereignty has been ceded. So here we are in Australia. This is we're going up to the mountains, and just to put things into perspective, Australia is a really large, wide continent in general terms. It's quite dry. It's much bigger than Switzerland, and the high elevation alpine areas of Australia are down here in the southeast. And this is the snow country, okay, which is about 5,200 kilometres squared on the mainland, and about 6,500 kilometres squared in Tasmania. And the combined area is only 0.15% of Australia as a whole, but that's about 30% the size of Switzerland. So it's a relatively large and important region nonetheless. These are ancient, eroded, rounded, soil-covered mountains. So there's no permanent ice, there's no permanent snow cover, and only a handful of remnant glacial features from the last glacial maximum. And because of this, we have an almost complete vegetation cover across the mountains, even on the highest peaks in southeastern Australia. Australian mountains aren't too dissimilar to mountain environments all over the world, and the growth of alpine plants is tightly coupled to local environmental drivers like the variation in soils and topography, aspect, exposure, local snow cover patterns and drainage as well. And this is why we get repeatable patterns in plant communities across alpine landscapes globally. So we get heathlands and we get grasslands and we get herb-filled meadows, snow bed, snow patch communities. And this makes alpine environments really variable and heterogeneous landscapes. And because there's such strong correlations and tight coupling between these environmental variables and the distribution of these plant communities and species, it means that they're particularly sensitive to changes in the environment. So mountain environments mean that that's a really great place to tackle questions about climate and try to understand how plants will respond to decadal scale changes, as well as more kind of rapid interseasonal changes, rare events, and climate extremes. In these landscapes, the timing of plant growth and development is intrinsically related to snowpack, okay? So the depth, duration of snow cover. And the timing of the snow season and how much snow covers the landscape is especially important because it determines when the plants can grow and how much water they have available to them. And so with climate change, warming temperatures is going to affect the snowpack, as do all the topographic features of the landscape in turn. And this is where in the landscape it's going to become really dry in summer, and soils will start to, dry out much lower than wilting point for plants. So climate warming and a contracted snow season, it means a later start to winter and an earlier spring. And these are some of just the key environmental changes that many mountain environments are facing. So snow in these landscapes is really a key driving factor for a myriad of ecological processes. So snow cover keeps these plants in the dark over winter, so thereby controlling photosynthesis, and also provides protection from frosts and strong winds. It insulates the soil as it melts in spring. It cues the beginning of the growing season. So plants at that time are getting stuck into photosynthesis and growth and flowering. And for those couple of months, snowmelt provides a really steady, slow release of water. Now, there's lots of interannual variability in snowpack. There's some big years, there's some really poor years, like what we had in 2020 and last year. So we're seeing a real contraction of the start and end of the snow season in Australia. There's some big years back in the 1980s, you can see over here. Even 2000 was a relatively big year, but you can see just from this kind of cool image that the start of the snow season is kind of becoming later, and the end of the snow season is happening earlier. So 20 years ago, the Commonwealth Scientific Industrial Research Organisation made some predictions about what the snow cover would be for 2020. And this would have been a slide that I used as a young researcher at a conference. And unfortunately, these predictions in snow cover have largely come true, especially for the lower mountains in the southeast. So snowfalls are declining, and there's a projected 30 to 70% decline in annual maximum snow depth relative to 1990 levels by 2050 at Falls Creek, which is a high place, and Mount Hotham in Victoria, under a low emission scenario. But under that high emission scenario, obviously it's even more drastic. So we're actually seeing these kind of changes coming into effect already. And this is what a really poor snow season looks like. So this is from 2006. I didn't actually go cross-country skiing that day. I had to carry my skis and walk out to where I was going. And back then in 2005, people were paying $100 to buy a lift ticket and ski or snowboard down a patch of artificial snow. And really, things aren't getting that much better. This was last year, peak season, August. This should be covered in snow, covered in people. And now they're charging about $160 for a lift ticket to go skiing at some of these places. So climate change is real, and the effect on snowpack is real, and it's happening now. But as ecologists, what we really want to know is how has the vegetation responded to these recent decades of climate warming and then reductions in snow cover. So since about 2003, with John Morgan and Iris Hickman at La Trobe University in Victoria, we've been surveying alpine summit vegetation across Victoria in a really systematic way, using 1 metre square quadrats spaced evenly over 15 summits around the southeast of Australia. And if you wait long enough in Australian ecosystems, soon enough a fire will probably pass through. And some of these summits were burnt in 2006, but we did manage to find all of our marker pegs, and we continued with the survey. So today I want to show you sort of some of the trends and what's been going on at these summits over the last 20 years or so. So Iris has recently collated and analysed some of this data set for her masters. So here she's split up the data into life forms. So what we've got here, we're looking at the change in percent abundance of graminoids on the left, herbaceous things, and then shrubs on the y-axis against time on the x-axis. And you can see there's some gradual changes, okay? Graminoids in the burnt sites tend to increase, the forbs, herbaceous things, roughly stay the same or decreased at the burnt sites there. But importantly, shrub cover seems to increase, and more significantly, more steep increase at those burnt summits. And Iris used a state and transition model to assess summit vegetation structure in response to fire and warming. And she defined a state as forb-dominated or graminoid-dominated or shrub-dominated. And a transition occurred when a state changed. So on unburnt summits, here, there was high stability of states over time, but some patches were becoming shrubbier. If you compare that to the burnt patches on these summits, they have lower state stability and the summits are becoming shrubbier quicker, but there were some transitions to graminoids. Okay, so it's becoming overall in Victoria, so the most southern part on the mainland, those alpine summits are becoming shrubbier and grassier. Meanwhile, in the Snowy Mountains in Kosciuszko National Park, which is a little bit further north, slightly higher mountains as well in New South Wales, and over a similar time period, we've got some permanent vegetation monitoring plots that form part of a globally replicated study. It's called the Gloria study, and they've been repeatedly surveyed since 2004 by various people, including myself and Catherine Pickering, and more recently, Catherine's PhD student, Brodie Verral. And we have these 5 summits. They're all nicely arranged down a ridgeline that form this really neat elevation gradient. It varies in temperature, varies in snow cover duration, and these summits are long unburnt as far as we know. So there hasn't been recent fires up there. And Brodie, for his PhD, recently collated all of the past Gloria survey data, and he's published that. And again, we see the percent cover of vegetation on the y-axis here has been increasing in general, all right? And we're seeing more cover of graminoids, not much change with forbs, but again, an increase with shrubs, all right? And when you compare across the elevation gradient of sites, vegetation in general is increasing through time as well, with probably a higher cover abundance at the lower elevation sites, with the lower elevation sites here on the far right here, okay? So again, shrubs are contributing to these increases. So just this graph on the bottom here, especially at these lower elevation summits where it's warmer, snow melts first in spring. So the plants there have a longer growing season. So something's definitely going on with shrubs, okay, in Victoria and in New South Wales. And we can use this natural environmental gradient of these 5 summits to then make predictions about vegetation change at the highest elevation summits. So using like this as a space for time analogy, whereby we imagine that the patterns that we see now at the lower elevation summits is what we might see in the future at the higher elevation summits. So essentially, we're probably going to see a higher abundance of shrubs and some more grasses. And this makes sense, right? We know that shrubs require more heat in the system to build and maintain their expensive woody tissue. That's why they're typically excluded from areas in the landscape that are really cold, okay? So like frost hollows, or where snow accumulates well into summer, like these kind of snow patch communities. So you can see these big dark green blobby things around the edge, they're all shrubs, okay? And they're not so common in the area where snow lasts later into the season. So like the elevational gradient of summits, we can use this natural experiment in snowmelt to also make predictions about vegetation change in these snowpatch communities. And it just so happens that across the Warby Corner snowpatch we call on the Bogong High Plains in Victoria, we've been keeping track of the snowmelt patterns within this particular snowpatch. So the red lines around here indicate the outline of the snow in 2020 that a student of mine mapped. And again, you can see some green blobs around the outside. So these are the shrubs around the edges of the snowpatch. And this is a really regular pattern of melt, okay? So while these annual dates might vary, the early melting zone, the mid melting zone and the late melting zone, they're in the same place year to year. And this still matches pretty closely with what I defined as early, mid and late snowmelt zones 20 years before, okay, during my own research project in 2000. So we've been resurveying these 217 permanent 1 metre square quadrats across this particular snowpatch community every. So I've just pulled this out of Ty's honours thesis. This is what he was focusing on, the abundance the y-axis. Here we've got years. We don't really going on between the first survey that I did in 2000 and then we did something in 2011. But again, in these the orange and red, this is the early and very early snowmelt zones. This is where we're seeing an increase in the percent cover of shrubs in these snow patches. So shrubs in particular, they're on the move, they're increasing in abundance, especially in the more favourable parts of the landscape where the snowmelt's earliest and where they're going to have a longer growing season. So for these special snowpatch plant communities, this particular assemblage of plants underneath those patches of snow, these are the specialist snowpatch species. They're threatened by a few things that are likely to result in that assemblage changing forever. So perhaps to the point of ecosystem collapse. And invading shrubs are on the list of threats. So we wouldn't be able to look at that landscape in summer and mark out where we thought a snow patch community would be, because just based on the vegetation, especially if it was really shrubby. So we can use tools like the red list of ecosystems, risk assessment criteria to help researchers and land managers sort of identify what the risks and what the threats to these ecosystems are, and then how they interact with each other. Because when you do a red list of ecosystems assessment, which we have done for all of the Australian alpine ecosystems, you get to make these cool conceptual diagrams to show how the various threats are affecting the characteristic biota that you're focusing on and how everything relates to each other. And then you get to set some thresholds and determine when you would consider that that ecosystem has collapsed and it's transitioned into a different ecosystem or a different community. So here, climate change up here has an indirect effect on snow cover via temperature and precipitation. Okay, and of course, shrub and tall grass invasion also have a direct effect on the snowpatch herb field. So these are the threats here in orange. And only a couple of months ago, John Morgan and Zach Walker, both at La Trobe Uni, published this neat little revisitation study, which analysed the vegetation in 7 snowpatches around the Bogong High Plains in Victoria. And guess what? Shrubs have been increasing in the majority of these snowpatches and essentially changing the assemblage of these plant communities into something else. So that we can't call them snow patches anymore. We have to call them something else. So they're more shrub dominated. And if the alpine landscape keeps becoming shrubbier, okay, with the common alpine shrubs from typical Aussie families like Myrtaceae, Proteaceae, Asteraceae, Fabaceae, Rutaceae, it means that the alpine landscape in general becomes more flammable, because most of these shrubs contain volatile oils in their leaves and they burn really, really well, okay? And they respond well to fire. So for example, a species, an Asteraceae shrub, Osothamnus hookeri's, common name is kerosene bush. So it's not a surprise that this is a kind of plant that burns well. So indirectly again, climate change is probably leading to our alpine areas becoming more flammable because of the shrubby thickening that's occurring. And the typical short alpine herb field type vegetation, characteristic of snowpatch plant communities here with fewer shrubs, it hasn't burnt so much as the surrounding shrubby landscape. Okay, so we can continue with our long-term monitoring. We can essentially watch the changing vegetation patterns occur in real time and make predictions based on past events. And we can also use environmental gradients as space for time analyses, so across snowmelt gradients, to suggest where the lower, warmer, earlier snowmelt areas could be and what to expect. So we can keep doing those things, and we probably will. Or we can also ask experts what they reckon, based on evidence of their own experiences. And this is what we did. We asked experts, expert alpine botanists, what they think will happen to the abundance of these snowpatch species through time. And a lot of snowpatch species are thought to decline. So here, species with a large proportion of orange colour are likely to decline over time. Species with blue might actually increase, and they could be species that exist elsewhere in the landscape as well. So yeah, if plants actually respond reasonably quickly to changes in snowmelt, we might not have to wait long until we see those changes predicted for 2020. Okay, so where does that leave us? I've talked about longish term changes, how alpine vegetation might respond over decades, but how are plants responding to their environment on a day-to-day basis? How will these responses contribute to the decadal scale changes? So I'm talking about these extreme events like heat waves and frost and drought and fire. And then ask how will plants recover if they're exposed to those sorts of events? Okay, so we know that winters are changing, but summers are changing too. And so to tackle these questions and tease apart and investigate the actual climate parameters that are changing and look at individual plant species and responses, we need to do some experiments. So that's where I'm going to leave my presentation today. And I would just like to acknowledge a couple of the folks that have helped me throughout my career. This is John Morgan from La Trobe. Also Ken Green, who was an original member of the GMBA many years ago up in Colorado, and also Adrian Nicotra at the ANU. And also thank all the students and postdocs and researchers that I've worked with over the last 20 years. That's made this so much fun. Thanks very much.
Thank you very much, Susanna. What an impressive talk. A very sobering talk as well. And I'm absolutely stubborn by how quickly things are changing and what the consequences are of those changes. You are talking about experiments, and that's where Jeronimo will take us now. Jeronimo, you were a PhD student of Susanna. You have brilliantly graduated. Congrats for that. And you are on the look for a postdoc. So if anyone out there has one for a brilliant scholar, please get in touch. You are an ecologist and a conservation biologist. You are also an excellent photographer. I had a look at your pictures, and I'm very much looking forward to hear now your presentation on the experiments you performed during your PhD. Jeronimo, the floor is yours. Thank you.
Thank you. Thank you all for attending this seminar. Can you see my screen now?
Yep. But yeah, yes.
Yes, thank you.
Yes. Okay. Well, hello everyone. Thanks for being here. Susanna just gave a quite impressive introduction to the Australian Alps and how climate change have affected the vegetation there. And I'm going to continue to talk about how plants respond to climate extremes. but I will focus on studies that instead of using long-term observations or natural gradients, we actually manipulated the environmental factors to simulate climate change.
You unshared your screen.
Yeah, it's sharing again, right?
Yes, thank you.
Okay, sorry about that. Well, I will give an overview of 3 different studies. All 3 focus on plant regeneration from seed under climate change. The difference between them is that where we manipulated the environmental factors. Like the first one, we applied the treatments in the laboratory and a greenhouse. In the second study, it was a totally field study. And the last one is a combination between field manipulation and laboratory treatments. Let's start with a greenhouse experiment. It's a salted bank experiment. In this study, we asked how the seedling emergence from the salted banks of 2 plant communities will be affected by climate change, and particularly by 3 changing environmental factors that are the reduction in the snow cover, fire, and spring summer drought. All 3 factors have changed and are expected to continue to change in the Australian Alps, as Susanna just told us. The main part of the methods of this experiment is characteristic of any other salted bank study, where we collected the soil in both of our plant communities, an Alpine airfield and a tree-line ecozone. We have different sizes to be sure that we were having enough replication, and then we also characterized the standing vegetation. The soil samples then were transported to the laboratory, where we dry them and we sieve them before do some cold certification and for some fire treatments. And after that, we put the soil in trays and in different water treatments in the greenhouse. But how we simulated climate change in this experiment? Well, to simulate one winter under the snow, we placed control samples in a freezer at 2 degrees for 10 weeks. To simulate a reduction in this snow cover, we reduced the cold certification from 10 weeks to 6 weeks. And during the other 4 weeks, we increased the temperature of the freezers to 5 degrees. And additionally, we simulate some frost events to mimic the lack of insulation of the snow cover, where we drop the temperature of the freezers at -5 degrees for 6 hours. And this was done for the first 3 days of every of those 4 weeks. To simulate fire, we put soils in oven at 80 degrees for 10 minutes, and then we add some smoke water to be sure that there were smoke chemicals in the soil. And finally, we had, through watering treatments in the greenhouse, a control where the soil metric water content was around 40 and 45, and spring summer drought where the soil metric water content was maintained around 30 and 35. This was a fully factorial experiment. I'm not expecting you to understand all these figures, but this is like a general timeline of experiment where we have the individual treatments, individual manipulations, and then all the possible combinations. The 40 weeks of the experiment, the first 10 weeks took place in the freezers, and the last 30 weeks took place in the greenhouse with the different water and treatments. What were the main findings? Well, here we have on the left side of the screen the seedling density plots, and on the right the emergence time, seedling emergence time. Let's focus on the left side, the seedling emergence, the seedling density. On the top we have the tree line, and on the bottom the airfield. Here is the number of seedlings that we recorded in each one of the treatments. Control conditions was where we recorded the higher density of seedlings. The snow cover decreased the Alpine airfield density for the airfield, the Alpine airfield, but not for the tree line. The fire did not have a significant effect on the number of emergent seedlings, and the drought have a negative effect or decrease the number of seedlings that we record in these drought trays. Drought also have a different influence in the seedlings. That was the emergence time, where most of the seedlings emerged after the week 20, whereas when we stopped we increased the water, soil volumetric water content to match the control conditions. Regarding the seedling the diversity that we found in the trace in the soil seed bank, these are two plots with diversity profiles for the two communities on the top airfield and in the bottom the tree line. Each one of these lines is a profile for one of the different treatments, but let's focus on the yellow one that is the control and is where we found the higher values of diversity. There's no treatment, that is this green one here next to the yellow, did not affect the diversity of the seedlings. However, fire affected the evenness of the seedlings. We can see it's not affecting too much the richness of the species that emerge from the soil seed bank, but when we start taking into account the density of the seedlings, this line drops more drastically than the control plots. And finally, drought here in the bottom of both plots reduce the richness in both communities. When we try to explain our findings with some seed traits, we were not able to do that. However, plant life forms help us. All the treatments increase the similarity with the standing vegetation. If you want to know a little bit more about this experiment, here are the details of the publication. Let's go for the second experiment. This was an in situ manipulative experiment where we asked how a future drier and warmer climate and post-fire conditions will affect germination and seedling establishment of 13 common alpine species in Australia. The study took place in the Victorian Alps, and we select 3 sites with similar environmental conditions, and in each one we established 12 plots. These plots were randomly assigned in equal numbers to different experimental conditions that were the result of a 2-factorial experimental design. Here is shown like future climate, control, fire, and future climate plus fire. Here is a closer look to how the future climate treatment was imposed. This is a warmer and drier microclimate. And we use modified open-top chambers for this. We took the traditional cone open-top chambers and we add these rain-out structures to create a warmer, like this thermal image shows, a warmer and a drier microclimate. The fire treatment was applied by burning selected areas of the experimental plots, and we used like a hand torch and a metal ring. We also add some smoke water to be sure that the smoke chemicals were present in the soil. Then we bury mesh bags with the seeds of our study species, and we plant seedlings in all the plots. And in 2 years, all the snow-free season, we came every month and recovered the mesh bags, record how many of the seeds germinated, and record survival and other characteristics of the seedlings. Finally, I'm going to talk about an experiment that is a mix between in situ manipulations and laboratory experiments. And here we ask how a drier maternal environment will affect the drought tolerance of the next generation seed and seedlings. This is the field manipulation consisted in rain-out shelters that are part of the Australian Mountain Research Facility. And after a year of these shelters being established, we collect some seed of 3 species, 2 forbs and 1 grass. Here is a plot showing the effect of the shelters in the soil volumetric water content. The yellow is the shelter plot, and the green is the control conditions. With the seed that we collected here, we performed some germination experiments where we exposed the seeds to a gradient of different water potential solutions that we created using polyethylene glycol. And then we also grew some seedlings and expose them to different watering treatments. Some of the main findings, here we have the effects of drier maternal environment. in the seed traits of our study species. Here is the 3 species, and here the seed size and the seed mass. We have contrasting results when the seed size and the seed mass of the 2 forbs are smaller under the shelter plots, and in contrast, the seeds from the graminoid species from the right heterosperma were larger under the shelter plots. However, all seeds responded equally when we expose them to different water potential solutions. In these plots, we have the final proportion of germinated seeds and the different water potential solutions. Here 0 is control, is just water, and here is negative values, is less water available for the seeds. And we can see how the seeds that came from the shelters, from drier maternal environments, is the yellow columns. These seeds have better capacity to germinate on drought conditions. The seedlings, similarly, the seedlings from dry maternal environment have a better tolerance to reduce water availability. This photo is super illustrative. In the left, we have seedlings from the dry maternal environment, and in the right, the ambient or the control plots and the different water treatments. And we can clearly see how the seedlings on the drier environment are larger, have more leaves, are more green. They have higher values of SLA, higher values of ABFM, and this photo was took after 6 weeks of the experiment. Before finishing, I just want to say that we should expect changes in germination time, seedling emergence time, seedling establishment, and the diversity of the seedlings being established as a consequence of climate change in the Australian Alps. And also that some of the versions that I have just shown are consistent or coincide with the reported results from studies that have looked at how annual variations in temperature and precipitation affect germination and seedling survival. And consequence, manipulative experiments are useful to improve our understanding of how alpine plants will respond to climate change. Ideally, we have a combination of manipulative experiments like these ones, long-term studies like the ones that Susanna showed, gradient experiments, to have a comprehensive understanding of how plants will respond and how what are the decisions that we need to take to preserve these environments. Finally, I just want to thank all the funders, the ESA Football Research Endowment, Deakin University for the financial support. Field experiments are cool, but are also like expensive. And also, I want to acknowledge the traditional owners of the land where I live and this experiment took place, and all the volunteers that are part of these field experiments.
Thank you very much, Jeronimo. I'm impressed as well by your presentation. Complements that of Susanna excellently. You're not only a good scientist and a good photographer, but your presentation was absolutely neat and elegant and clear. So thank you very much. No doubt, whoever hires you as a postdoc will have taken the right decision. We are moving on to another biome now, 2 brilliant women scientists from Austria, although Sabine has now decided to go up north to Uppsala. Manuela Winkler is working in Vienna at the University of Natural Resources and Life Sciences, where she's working on plants and also fulfilling the role of scientific coordinator of the Gloria program. And she worked hand in hand with Sabine on some question of genetic diversity, and I think that works perfectly well with what we've seen before, zooming further into the details of how vegetation reacts to climate change. Manuela and Sabine, the floor is yours.
Yeah, thank you very much. I will just try to share my screen.
This is your email, though.
Yeah, I know.
Here we go.
Now,
perfect. Thank you.
You now see the relevant part, I hope.
We do.
Yeah, that's
Thank
great.
you.
Yeah, thank you very much for inviting us, and good morning and good evening to everybody who is listening. So as Stavna already said, we are moving now into the intraspecific dimension of biodiversity in the context of climate change. And it may sound a bit trivial, but we already saw it in or heard it in Jeronimo's talk. The individuals belonging to a species are not all the same. So you have intraspecific genetic diversity, and that's what we are going to talk about today. And this genetic diversity is sort of the raw material for evolution and local adaptation. So evolution shapes species responses to climate change. But on the other hand, climate change also affects evolution by imposing selective pressures, by causing mass mortality from extreme events, or by disrupting gene flow. And this again has consequences for species responses, and this creates some sort of eco-evolutionary dynamics. We can tackle these things, or these climate change, intraspecific genetic diversity dimension, roughly with 3 approaches. So we also have space for time analogies, species distribution modeling, but also genetic monitoring. And we're also going to look into challenges associated with this and future directions. If we're talking about space for time substitution or analogy with regard to intraspecific genetic variation, a landmark project was the IntraBiodiv project, which took place between 2004 and 2006, which was led by Pierre Tabalé. It was a multidisciplinary EU project. And in this multi-species study, we looked at intraspecific genetic variation in relation with species and habitat diversity across the Alps and the Carpathians. And one of the key results of this study was that genetic diversity does not always coincide with species richness in alpine plant communities. So that is to say, if you have a high species richness somewhere, this does not mean that genetic diversity will also be high, and vice versa. And 20 years later, we did another space for time analogy study. And please, Sabine, maybe you can tell us more about this one.
Yes, so I will just use your screen, I guess.
Yes, please.
Okay, so in our study from last year, we essentially looked at intraspecific genetic diversity of 2 snowbed species, Achillea clusiana and Campanula pulla, here shown. And they are both endemic to the northeastern limestone Alps in Austria. And we sampled them from the lower, middle, and upper elevation range on these 3 mountains here, ranged from west to east. And we found a high proportion of private alleles in populations that are in the lower range, and they are the most threatened by climate change because the tree lines are moving upwards. So these private alleles, they are in risk to being lost because of habitat loss due to climate change. This is reducing the local population sizes and causing bottlenecks. But bottlenecks are also caused by populations moving upwards to higher elevations. So these private alleles occur only in these populations, and the genetic information is lost in case these populations disappear. So if we go to the next slide. Yeah, I think, yeah. We further had some environmental data that we also analyzed. So we had some metadata on elevation, precipitation, eastness and northness, as you can see here in these redundancy analysis plots. Eastness and northness are also both highly correlated with like sunlight or wind exposure or general weather conditions, also temperature. And in an environmental association analysis using redundancy analysis, we showed that some of the alleles are actually associated with climate-related variables. So they harbor adaptive potential. And there's, so to speak, some kind of race between climate change and migration and how fast these rare alleles can actually spread and rise in frequency and be maintained in the populations. So that is, why is that so important? Because of their adaptive potential, which is needed in the face of climate change. And I think now Manuela will take over again and give a little bit about insights into what is relevant to understand migration dynamics with this modeling.
So one possibility to look at how this spread may happen are species distribution models. So we have traditionally those models are static ecological niche models. So you just relate the bioclimatic variables to species occurrences. But more recently, more dynamic ecoevolutionary models have been developed that combine those niche models with individual-based demographic and genetic simulations. Mind genetic simulation so far, at least for Austrian or Alpine plants in general, because there's not so much real data yet. And for 4 endemic plant species of the Austrian Alps, these simulations show that in the dynamic models, perennial species persist in unsuitable habitats longer than predicted by the static niche models. Those are given here in red. And this causes delayed range losses. But the long-lived adults that persist in those unsuitable habitats produce increasingly maladapted offspring, and this leads to sharply decreasing population sizes in the long run. But if we really want to know what is happening, we need to look at changes over time, and here genetic monitoring comes in. So what do we understand under monitoring? These are repeated surveys in which qualitative or quantitative observations are made, and they usually use a standardized procedure. If we're talking about monitoring in the context of climate change, we also have to mention GLORIA, the Global Observation Research Initiative in Alpine Environments. This was initiated around the turn of the millennium, and it now encompasses about or more than 130 regions on all populated continents, and we're covering 3 fundamental gradients: latitudinal, longitudinal, and altitudinal, and also temporal gradients. And but from the essential biodiversity variables for monitoring, we are mostly covering the species dimension. So we are looking at community composition, taxonomic diversity, species populations, and species traits. But we also plan to go into genetic composition, as I will show you later. But for these genetic composition, there were also proposed more detailed genetic composition essential biodiversity variables by Sean Hoban et al. Only 2 years ago. And these genetic composition EBVs are genetic diversity. So this is the classical richness or heterozygosity, but also genetic differentiation. So the number of different genetic units, the distance between them. Also inbreeding and the effective population size, which is roughly the number of breeding individuals, so those individuals that actually contribute to the next generation. If we look now at examples for genetic monitoring of alpine plants, there is not very much going on, at least to my knowledge. We have the National Biodiversity Monitoring in Switzerland, and they have recently completed or are about to complete a pilot study for monitoring of intraspecific genetic diversity. And this also includes, apart from some animal species, those two plant species. They have a very large elevational gradient, which also reaches into the alpine zone. They have a Swiss-wide sampling design, and they're using the latest hot shit, let's say, in sequencing technologies. This is whole genome sequencing. But in Austria, we are also about to start a project, a genetic diversity assessment of plant species endemic to the northeastern limestone Alps. Here, two of our species that Sabine was talking about before, Achillea clusiana and Camadula pulla, are also represented. And here we are going to have a range-wide representative sampling, as they are endemic to the small northeastern limestone Alps. And we are looking at this genetic diversity using restriction site-associated DNA sequencing. So genetic monitoring is associated with quite a few challenges, logistic and technological ones. And this starts with field collection. So you have to decide which species do I take, where do I sample, how many individuals, how often do I repeat the sampling, and this also has to be related to the generation time of the species I select. Then in the lab, I have to decide the method of with which method do I extract the DNA. I need to choose a genetic marker, and also have to keep in mind that technology is always advancing, and that maybe in 20 years or 10 years when I'm resampling, that there will be other technologies or better technologies available. I have to choose a sequencing platform. And then, of course, a very important part is the bioinformatics part, where I look at the data generated with sequencing, and of course, how exactly do I analyze the data. And Sabine is now going to delve a bit more into the technological issues associated with sequencing. Sabine, please.
Yes, so if we look at the next slide, we're gonna delve a little bit into the pros and cons of two sequencing technologies that we decided to explain a little bit more in detail. So we have RADSEQ, the restriction enzyme-associated DNA sequencing versus whole genome sequencing on the right side. So as the name already says, RADSEQ is using restriction enzymes. So it makes use of restriction enzyme cutting sites along the genome, which are displayed here in this with these black bars. And it essentially cuts, depending on what enzyme you have, it cuts the genome into fragments. And then you ligate some adapters, and then you select for a specific fragment size, depending on what sequencing allows. You add the second adapter to it, and then you essentially have this, after sequencing, you have these stacks of reads that are mapping to parts of the genome. So it's just a reduced representation of the genome. While on the other hand, whole genome sequencing just fragments the whole genome, the molecule, into random fragments. You add the adapters, you sequence, and then you have like a read pairs covering more or less the whole genome. So if you have a certain budget, essentially you get, with using RADSEQ, you get more depth for only a part or reduce the representation of the genome, but you have greater confidence in the genotype calls. and also a greater number of samples that you can analyze for the same money compared to whole genome sequencing. What also comes with the data is RADSEQ is more easy to handle. Like it needs less storage, it needs usually less capacity, like computational resources to analyze the data, but also the tools are easier, or that you don't need so much bioinformatics knowledge and not so much customized pipelines to actually analyze the data. I think we can show the next points now. Yes, so RADSEQ is suitable if you want to do a pilot study on something, on an organism that is not a model, you don't have a reference genome yet or no prior knowledge. You just want to see, is it worth to dig deeper, or to show that it is worth to dig deeper into this species and analyze it in more detail. But still you can get first insights into population genetics data. While whole genome sequencing is nice if you have a more advanced study, you have a reference genome already that you can use to compare all your samples and to find variation. And it gives more refined estimates also for population genetics. And it allows to study adaptation and selection in more detail. So as to exemplify this, as Manuela already said, we were using RAD sequencing for our study last year. And based on the variants that we found there, we were able to have first insights into population genetics, like admixture profiles here shown with these colored bars for the different mountains from west to east. So you can see that the Kaibling, for instance, has a very private admixture profile. And then you see a little bit of mixture with the Hochschwab. And then the Schneeberg is a super admixed population there. Or we could also combine it with the environmental variables that we have, but we've seen this before already. And if we look now down here in this plot that now appeared, we can see an example where we used whole genome sequencing, or colleagues in my lab where I'm currently working in Uppsala, whole genome sequencing of herring populations. And covering the whole genome, it was much easier to detect variants that are associated with, in this case, adaptation to salinity or temperature or spawning. And when you look at the smaller plots here, so on the x-axis, you see along the chromosomes, the variation that is found. And you see that there's these high stacks, which are corresponding to inversions that occur in these chromosomes. And they were associated with the adaptation to, whatever variable was looked at. So this is really hyped at the moment that you look at inversions that are relevant for adaptation. And this is possible due to whole genome sequencing, because you have really covered the whole genome, and you see long regions that are connected to each other. So one thing that plays a major role, of course, is the money. So there are several constraints that people are facing who want to conduct, especially biodiversity studies. And on top of it, of this pyramid, there's money. Other things are, for instance, also policies for monitoring, but also coverage in the sense of taxa that are analyzed, or also, thanks for going back, the spatial coverage of the samples that you have. And one thing that I always find amusing is that the lack of human ability to actually analyze the data is, we lack that more than data that is available. So one thing which would be a nice idea is, or what people are doing, is to actually come up with project ideas that make use of data that is already available. But that's maybe a little bit tricky for biodiversity studies, but it's an option. And now we can look at the table where we see the really big difference in what the costs are for RADSEQ and whole genome sequencing. You can read the numbers yourself, I guess, so the difference is big. Just a comment, low coverage means about like a coverage of 2 along the genome. High coverage is above 15 or even more. So the high coverage data can be used to generate reference genomes, which are, yeah, one of the key tools for whatever analysis you're doing. Yeah, and I think now we can go to some concluding remarks from Manuela.
Yeah, I'd just like to add,
yeah.
so this is just the lab costs, so only for the consumables and sequencing. So you haven't done any sampling, you haven't paid the bioinformatician. And if you take as an example the Swiss genetic monitoring, where you have 60 populations and 10 individuals each. So you have only for these lab things, and you haven't even extracted the DNA, between 10 and 40,000 euros. So this is really expensive. And financial constraints are, I think, here even more acute than they are for, say, species level monitoring or biodiversity studies. And yeah, if we are looking at the future of genetic monitoring in the context of climate change in alpine environments, we plan to establish interspecific genetic monitoring of alpine plants covering the entire species distribution areas within Gloria. And I think this is really unique as far as I know. And we are now more or less in the conceptual stage where we are looking or trying to conduct baseline surveys in model species and determine adaptive alleles in these model species and develop guidelines. And then in a second and third step, we'd like to implement this first in Europe and then also globally within Gloria. Yeah, I also like to thank the Gloria team and all the people who helped in the field. And thank you for your attention.